Hello, my name is Veronica Masters from Eisenhower High School, and my science fair project is Effect of Temperature on Hydrolysis of Bromelain. Enzymes are protein catalysts that affect the rate of metabolic reactions when the active site is bound to the substrate, lowering the reaction energy and increasing the reaction rate. A factor that affects the reaction rate is temperature, as higher temperatures increase the speed of molecules and the collision of enzymes and substrates. Proteolytic enzymes through hydrolysis catalyze the breakdown of proteins by decomposing the ends of the peptide bonds with water. An example of this type of enzyme is bromelain, which is found in pineapple and provides health benefits in digestion. Although bromelain is stable at room temperature changes can affect its effectiveness, such as when it reaches 8 degrees Celsius. When bromelain denatures, the rate of reaction decreases because of the loss of shape, which the substrate can't bind to its complementary active site. Consequently, the denatured bromelain loses its effectiveness because of the ferric collisions. Suppose the enzymatic activity of bromelain can be observed at specific temperatures. In that case, this knowledge can be applied to find the best temperature the pineapple can prepare to ensure bromelain's effect. This will be done by measuring its enzymatic activity through the hydrolysis of the substrate gelatin that coats photographic film. When gelatin is catalyzed, the mass of the photographic film decreases as the gelatin is destroyed, showcasing bromelain's effectiveness. The rationale of this experiment also tests distilled water as a control variable for the hydrolysis reactions, which could break down gelatin. The pH of pineapple juice was stabilized using a pH buffer as another control variable to test if the acidity of the pineapple juice could be a factor in breaking down gelatin. Additionally, consuming acidic foods like pineapple have potentially negative effects, such as changing the pH of the body's waste products. The research questions include: what is the effect of temperature on the enzymatic activity of bromelain, and is there a significant difference among the 5 temperatures bromelain is exposed in the hydrolysis of gelatin? The hypothesis is the enzymatic activity of bromelain will increase in a positive linear relationship to this temperature. The optimum temperature of bromelain is between 50 and 60 degrees Celsius. Therefore, as the temperatures increase, so to that range, so will bromelain's effectiveness. The independent variables include the different temperature conditions the bromelain enzyme will be exposed. The dependent variables include the rate of change of the film strip's mass and appearance of film strip. The controlled variables include the film strip's original mass, duration of the experiment, and the volume of pineapple juice, stabilized pineapple juice, and distilled water. The materials include the analytical balance scale, photographic film, 50% pineapple juice, pH-free buffer solution, distilled gloves, incubator, ice bath, blender, tray, pipette, scissors, plastic containers, ruler, knife, strainer, and timer. To prepare the pineapple juice, first, using a knife, trim the top of a whole pineapple. They cut a section parallel to the trimmed top into chunks. With a digital scale, weigh 20 grams of pineapple chunks and place inside a blender, alongside 20 milliliters of distilled water to dilute the pineapple juice to 50%. Then blend until the mixture is a smooth liquid and then strain the pineapple juice. Finally, collect the pineapple juice inside a beaker and stir to create uniform enzyme concentration before pouring 8 milliliters into each labeled plastic container using a pipette. There will be 5 plastic containers to conduct the 5 trials. To prepare the film strips, remove the film from canister and cut 5 pieces measuring 1 centimeter wide while wearing gloves to prevent hand oil on the film surface. To prepare the distilled water, measure 40 milliliters with a digital scale and pour 8 milliliters into each of the 5 labeled plastic containers. To prepare the stabilized pineapple juice, 12 milliliters of pH-free buffer was measured with a digital scale and combined with 30 milliliters of fresh pineapple solution. Then 8 milliliters of the solution was poured into the 5 labeled plastic containers. After preparations for the experiment were completed, the procedure detailed below was followed. Step 1. Measure the mass of the 5 photography strips with a digital scale. Step 2. Appropriately label the 5 pineapple juice containers and insert the 5 photography strips, making sure to secure the lid. Step 3. Place the juice containers in the corresponding temperature conditions using an incubator or ice bath for 10 minutes. Step 4. After 10 minutes, remove the containers from the incubator/ice bath and measure the new mass of each film strip. Step 5. Repeat steps 1 through 4, replacing the 50% pineapple juice with stabilized pineapple juice solution and distilled water. Repeat step 5 for the 5 different temperature conditions tested. This is the experiment setup with the 5 plastic containers, film strips, substances, and plastic tray. And this is the qualitative data which demonstrates the film strip's appearance before and after the experiment. When the gelatin is hydrolyzed, the silver grains fall off the film, so there's nothing binding them to the plastic backing. And this is the quantitative data for all the 5 different temperatures and substances the film strip was exposed to. The initial mass, final mass change in data, a percent change was found for 0 degrees Celsius, 10 degrees Celsius, 26 degrees Celsius, 4 degrees Celsius, and 6 degrees Celsius. Furthermore, a data analysis was conducted on the percent change means for the different temperatures and substances in addition to the standard deviation, variance, and mean. When the ANOVA test was performed, a significant difference in the mean percent changes for all 3 substances was found, with the F value 3.89 being greater than the F critical value 3.88. And this is the visualization of the mean percent changes of the film strip's weight throughout the trials. Further analysis utilizing Tukey's test revealed a significant difference between the solutions for bromelain present in distilled water solutions for the 5 treatments. Using the critical value 9.19, a significant difference was found for stabilized pineapple juice at 26, 40, and 60 degrees Celsius, while a significant difference for pineapple juice was only found at 40 and 60 degrees Celsius. These findings suggest that temperatures near the optimal range of bromelain, 50 to 60 degrees Celsius, significantly increased bromelain hydrolysis. Additionally, the stabilization of pineapple juice could have increased the efficiency of bromelain hydrolysis, with a significant difference at 26 degrees Celsius for the film strips exposure to stabilized pineapple juice. When exposed to pineapple juice and stabilized pineapple juice, the film strip's mass increased the greatest negative percentage mean of negative 10.1084 and negative 5.68 at 60 degrees Celsius, demonstrating that bromelain hydrolysis of gelatin decreased the film strip's mass. Both the ANOVA test and Tukey's test revealed a significant difference in the mean percent changes for stabilized pineapple juice at 26, 40, and 60 degrees Celsius, and pineapple juice at 40 and 60 degrees Celsius. Meanwhile, the distilled water control experienced a positive percentage mean for all temperatures, showing the film strip's mass increase. The data collected shows temperature's effect of bromelain hydrolysis with exposure to 40 and 60 degrees Celsius, increasing the rate of reaction, while exposure to room temperature 10 degrees Celsius and 0 degrees Celsius, decreasing the rate of reaction for bromelain. And the limitations or recommendations include one of the pineapples utilized at different ripeness, which may affect the type of bromelain utilized. The incubator wasn't sealed completely, which would have allowed for temperature fluctuation, impacting the rate of hydrolysis of gelatin. Further experiments should be conducted in a completely sealed incubator. The 10-minute trials could have slight time differences due to the time it took to remove the film strips from the solutions. This could change the time allocated for hydrolysis and impacting the protein strip's mass. These are the references utilized in this experiment.